The overall goal of this procedure is to form cancer cell spheroids in a standard high-throughput format in order to use them in anti-cancer drug testing. This is accomplished by first dissolving two polymers, polyethylene glycol and dextran, in cell culture medium at predetermined concentrations to form a successful aqueous two-phase system. The second step is to prepare the cells of interest at twice the desired cell density and mix the cell suspension with an equal volume of the aqueous dextran phase. Then, fill a 96-well plate with the aqueous polyethylene glycol phase and dispense sub-microliter droplets of cell-containing dextran phase drops into the wells using a liquid handling robot. The next step is to confirm spheroid formation in the 96-well plates after 24 hours of incubation and to directly add anti-cancer drugs of interest to the wells. Finally, a standard plate reader compatible viability assay is used to show the percent viability of drug-treated spheroids normalized against control non-treated spheroids.
The main advantage of this technique over existing methods, like the hanging drop method or the microfabricated well approach, is that it does not require customized plates or devices and allows high-throughput spheroid formation and drug testing. This method can help answer key questions in cancer research and expedite discovery of anti-cancer chemotherapeutics.
The implications of this technique extend towards the therapy of cancer because it allows high-throughput screening of chemical compounds with more relevant tumor models to identify new anti-cancer drugs.
Individuals new to this method will conveniently form cancer cell spheroids in micro well plates and will easily conduct drug treatment experiments using standard lab